Hello everyone. Today we are going to start with a new topic, that is embryology and the anatomy of the inner ear. So first of all, let's understand that the inner ear consists of a membranous labyrinth surrounded by a bony labyrinth. Okay, as you can see in this diagram, this one is the outermost part, it is the bony labyrinth, and this one is the innermost, it is the membranous labyrinth. And inside the membranous labyrinth, there is the endolymph, and surrounding the bony labyrinth, there is the perilymph. Okay, now, as we can see that the membranous part of the inner ear, it is also called as the membranous labyrinth, okay, which develops from an area of surface ectoderm. Okay, now if we see that, first of all, let's see the development of it. So it develops from the otic placode, okay, as you see in the this diagram, and then after it, that is, this becomes the otic pit and then the otic vesicle. Okay, guys. So the membranous labyrinth, it is like a balloon. Okay, now first, what does the what does it contains? Okay, membranous labyrinth. So as you can see in this diagram, there are three semicircular canals. One is lateral semicircular canal, one is superior or anterior super semicircular canal, and one is the posterior semicircular canal. And below to it there is the utricle, and below to it there will be saccule. Okay, and after that there is the cochlear duct. Okay, now the three semicircular canals, they are at an angle of 90 degrees to each other. Okay, now in the this one is the utricle. The three semicircular canals open into the utricle by the five openings because of the crus, which is the part of an anatomical structure. Okay, the posterior and the superior semicircular canal fuse together to form this one is the posterior and the superior semicircular canal. This fuse together to form a crus communes, that is the common opening. Okay, and which all together opens into the utricle. Now what is the saccule? So the utricle, it is connected to the saccule through a utriculosaccular duct. This duct, that is the utriculosaccular duct. Okay, this forms the endolymphatic duct, which ends blindly into the endolymphatic sac. This one is the endolymphatic sac, which is formed by the ducts of the saccule and the utricle. It absorbs the endolymph. Okay, and the endolymphatic sac, it is responsible for the absorption of the endolymph, and the endolymph is a fluid that fills the whole of the membranous labyrinth. Okay, now below to it, it is the membranous cochlea part. So after the saccule, it is the membranous cochlea, and the saccule is connected with the cochlea by the ductus reunions. So here, as you can see in this diagram, that the saccule is connected to the organ membranous cochlea by the ductus reunions. Okay, and the membranous cochlea, it is also known as the scala media. So the membranous cochlea or the scala media, it is a coil tube which takes 2.5 to 2 and 3 by 4 tons, 2.5 to 2 and 3 by 4 tons around a bony axis. Okay, so the sensory, this organ of Coti, the sensory organ of hearing, it is known as the organ of Coti, and the organ of Coti, it is situated in the basal turn of the membranous cochlea. Now the basal turn, it is responsible for the high frequency sound. Okay, and whereas the organ of Coti is situated in the apical turns. So in this diagram there is, first, as we can see here the diagram, first is the basal tone, which is responsible for the sensing of high frequency sound, and the apical one, it is responsible for the sensing of low frequency sounds. So therefore there are diseases which affecting the apical tone will lead to the low frequency hearing loss, whereas those affecting basal tone will lead to high frequency hearing loss. as we know that in the Meniere's disease, which is characterized by the increase in the endolymph, the narrow apical part of the scala media experiences the dilatation first, therefore affecting the low frequency sounds first. Okay. Now, as we can see here in this diagram, there is the bony labyrinth. Okay. Now in the bony labyrinth, the embryology part, it develops from the mesoderm by the endochondral ossification. Okay, the what is the enchondral ossification? That is the cartilage which is developed from the bone. Okay. Now there are some points like what the structures which are fully developed at the birth are. There are like the tympanic membrane, medullary cavity with the ossicles, mastoid antrum, bony, and the membranous labyrinth. So now the development of the bony labyrinth, as you can see here, there are three semicircular canals, the vestibule and the bony cochlea, scala vestibuli, and the scala tympani. So, the three bony semicircular canals, it is the bony labyrinth around the membranous semicircular canals, and the vestibule, it is the bony labyrinth around the utricle and the saccule. Okay, now the bony cochlea, scala vestibuli, and the scala tympani. So here the portion of the bony labyrinth are on the two sides. Okay. Now this, as you can see here in the diagram, it is the part of the scala media, and the above to it there is the scala vestibuli, and the below to it that is the scala tympani, and it is the turn which is known as the helicotrema, which connects the scala vestibuli and the scala tympani. Okay. So now we can we can see the diagram of the organ of Corti here. So as we can see in the diagram, that is the organ of Corti, it is present in the or scala media part, which divides the bony labyrinth into the scala vestibuli and the scala tympani. And as we discussed that it takes 2.5 to 2 and 3/4 turns. Okay. So now as we can see in this diagram, so there are two membranes. Okay, here we will discuss the two membranes such as the Rischner's membrane and the Bacillus membrane. So the basilar membrane, it separates the scala media or the cochlear duct from the scala tympani, and the Rischner's membrane, it separates the scala media from the scala vestibuli. So in here we will also see one more structure, that is the stria vascularis. So here there is one stria vascularis. As you can see, it is the perilymph and the endolymph structure. Here the scala media, and it is the scala vestibuli part. So now here this what what is the what is the benefit of the stria vascularis? So it is a site for the production of the endolymph, and it also plays an important role in the cochlear homeostasis by generating the endocochlear potential and maintaining the ionic composition of the endolymph. So here in this travascularis, it has two channels, which is sodium potassium and the sodium potassium 2 chloride channels, which is responsible, as we also know that it is the same channel which is present in the loop of Henle in the nephronic tubule on the on which the loop diuretics act. So what will be the endolymphatic potential? It is plus 80 to plus 85 millivolt. Now as you can see the organ of Corti, there are the structures. In the structures there are hair cells, some of the supporting cells, and some of the tunnel of the Corti. So there are inner hair cells and the outer hair cells. Okay. So the two types of the hair cells in the organ of Corti, the inner and the outer, they project into a membrane which is known as the tectorial membrane. As you can see in the here, this one is the tectorial membrane. Okay, guys. So what are the supporting cells? Supporting cells, they support the outer hair cells, which are also named as DCH, like Dieters, Claudius, and Hansen. Now here we will also see the difference between the inner hair cells and the outer hair cells. So in this chart we can see that there are, first, on the basis of rows, that the inner hair cells are arranged in a single row, whereas the outer hair cells are arranged in 3 to 4 rows, and inner hair cells are less in number and more resistant to the noise, trauma, and ototoxic drugs. Okay, but whereas the outer hair cells are much more in number and more resistant to trauma and the ototoxic drugs. And the nerve supply of it, so the nerve supply, it is mainly afferent of the inner hair cells, and the outer hair cells mainly efferent. So first, as we know, what is the function of the inner ear? It is the transduction of the sound, that is, to conversion of the mechanical sound into the sound energy into electrical impulses. So here we will see the auditory pathway. So first, as we start, that the inner hair cells stimulate the afferent nerves, which fuses to form the cochlear nerve, which transmits the spiral ganglion. This is the cochlear nerve. Okay, and from where the cochlear nerve exits through the internal acoustic meatus. Okay, so here the mnemonics can be slim, like it can be, first of all, the S for the superior olivaris complex, and L for the lateral lemniscus, and I for the inferior colliculus, and I for the M for the medial geniculate body. So from the medial geniculate body, the auditory fibers pass through the posterior limb to the internal capsule. Okay, so here we will see how the transduction of sound occurs. So first of all, that the opposite movement of the scala vestibuli and the scala tympani. Okay, so which creates a shearing action which leads to the movement of the basilar membrane, which leads to the stimulation of the organ of Corti, which contains the hair cells. So here, from the tip of the hair, outer hair cells have stereocilia, which is touching the tectorial membrane, and the tectorial membrane, it is the static one, which causes the movement of the stereocilia in the direction of the highest stereocilia. So such as the potassium channels on the tip of the stereocilia are opened, and the potassium from the endolymph enters the outer hair cells, so that there is the opening of the voltage gated calcium channels, and there is the release of the glutamate, which is a excitatory neurotransmitter, which is picked up by the afferent fibers. Okay, thank you, guys.